All right, so we are now talking about, uh, sorry, um, membrane fluidity and how a cell can maintain the fluidity of a membrane in a changing environment. All right, so if you think about it, you can go into the hottest weather in the Philippines, and most of the time, your cells are fine, right? They don't get leaky, you don't start losing the contents of your cells, and as long as um, as long as you don't get too cold, you can be in freezing temperatures for certain periods of time without um, your membranes freezing. Although, if you do um, stay out long enough, your cells will become damaged and die. But life can persist at freezing temperatures, and the reason for this is because cells can change their transition temperature. Okay, a transition temperature of anything. Okay, this isn't just for membranes. This is for liquids and water or alcohol or whatever. The transition temperature is the temperature where a substance goes from being a liquid to being a solid. So it's that temperature of phase change. If you think about water at normal conditions, the transition temperature of water is 0 degrees Celsius. Okay, so that is the temperature where liquid water becomes solid. Um, Now, you can change the transition temperature of water by doing things, right? You can add antifreeze to the water, and now, or you can even add salt to the water. And now the temperature that water freezes at becomes lower, right? So you can go all the way down to -10 and you still have liquid water. It hasn't frozen because you've altered the transition temperature, all right? Or you can do other things to it. You can add other chemicals to it, and you can increase the transition temperature of water so that you can be 10 degrees and you still have ice. You still have solid water. It hasn't melted yet. Okay? So if you change the transition temperature of water, you can change the temperature at which it freezes. So when you decrease a transition temperature, you decrease the temperature at which it will freeze. So if you decrease the transition temperature, you make something more liquid, okay? It's more fluid. It stays fluid at lower temperatures. If you increase the transition temperature, right, like you did to water here, then you're making something more solid. So now at higher temperatures, you have a more solid substance. Okay? Now, when we apply this to membranes, this is what the cell has to do to maintain the proper viscosity of the membrane. So if the temperature drops, if the temperature gets colder, the membrane needs to stay liquid. If the membrane freezes, so sorry, I should have gone back here. If the membrane freezes, it becomes uh, a crystalline structure that it can't keep the contents inside. It's no longer what do you say? It's lost its integrity. Okay, and so this is why if you've ever been in a place where it's frozen, the first freeze of the year, all of the plants turn into a gloppy mess because all of the cells in their leaves have lost the contents. Okay, this also happens because it forms ice crystals in the extracellular matrix and sucks out all the liquid. Um, but the membranes that are frozen, they can't maintain the integrity of the cell. Okay, so so the cell must do things to maintain the proper viscosity. So in order to keep a um, a more solid membrane, so in the Philippines, right, when it gets really, really hot, you have to make sure that the membranes don't melt, right, like butter becoming liquid. So the the cell can include more saturated fatty acids, or it can include, so if you have more saturated fatty acids, then the fatty acids pack together tightly like this, right? They pack together tightly and they can't move so well. If there are higher levels of saturation, there are lots of kinks in the fatty acid tails, which make more spaces between the molecules. So if there's more spaces, there's more freedom of movement. Uh-oh, what happened? Okay, sorry. So with higher levels of unsaturation, with more unsaturated fatty acids, you have more space between the molecules. They can move around more freely. So you make the membrane more liquid, you lower the transition temperature. To make it more solid, because the temperature is increasing, you make more saturated fatty acids. They pack together tightly, and the membranes, the molecules can't move around so freely. All right? Longer chain fatty acids prevent the movement of the molecules. So if you look at these long chain fatty acids, they're overlapping with each other and they're not going to move freely. So if you put your fingers together like you're going to hold your hands, think of those as long chain fatty acids. You can't move your hands because your fingers are overlapping. But if you make your fingers shorter, so they're not overlapping each other anymore, now your hands can move freely, okay? Because there's no overlapping of your fingers. So more movements. If the fatty acids are overlapping with each other, they're going to bonk against each other and they won't be able to move freely. Now, cholesterol maintains an intermediate fluidity. So cholesterol, when the temperature gets higher and the molecules want to move a lot, become too liquid, the cholesterol interferes with the mobility, right? Just like the overlapping fatty acid, they get in the way. The molecules can't move around so much, and so it makes it more solid. But when the temperature starts to drop and the molecules begin packing together tightly into a crystalline structure, then the cholesterol interferes with the packing of the molecules. And so it prevents the membrane from getting too liquid. So cholesterol doesn't just make the membranes more solid, it also makes them more liquid. And that's going to depend on the change of the temperature. It maintains an intermediate fluidity. All right. So be sure you understand transition temperature. If I ask a question that says, if the transition temperature increases, what effect will that have on the membrane? You should be able to tell me that the membrane will be more solid, right? If I say, if the transition temperature increases, or you should be able to also tell me something like, what would the cell do to increase the transition temperature? Okay, so then you have to think, well, if it's increasing the transition temperature, it needs to make the membrane more solid. What do I need to do to make the membrane more solid? Right, then you go here and say more solid, more saturated fatty acids, longer chain fatty acids. Okay? Okay. All right, so this is pretty much what we just talked about. I do want to mention that synthesizing new fatty acid takes a long time, okay, relatively. But the cell can very quickly change the transition temperature by remodeling existing phospholipids. And so there's several ways to do that. It can create single or double bonds within the existing fatty acid using desaturases and saturases. It can also change the phospholipids between two fatty acids. So it can take an unsaturated fatty acid and switch it with a saturated fatty acid on a different phospholipid. That will make a phospholipid with two unsaturated fatty acids, and that creates a very large kink. It creates a molecule with two kinks, which makes the molecule very, they won't pack together well at all. So it'll make the membrane more liquid. All right, what's going to make the membrane more fluid? Right? Shorter fatty acids, because they're not overlapping with each other, so they have freedom of movement. Okay? What's going to decrease the transition temperature? Right? So now you have to think. If I decrease the transition temperature, right, then at a colder temperature, I'm still liquid. So I'm making it more liquid. So to make it more liquid, shorter fatty acids. Okay? All right, let's talk about, so that was talking about the movement of lipids. Now I want to talk a little bit about the movement of proteins. So people weren't really sure. Before the fluid mosaic model came to its full maturity that it is at today, no one was really sure what proteins were doing. A lot of people thought that the proteins were static in the membrane. They couldn't move. And then this experiment, this very elegant experiment, was done where you had mouse cells. Well, you had proteins from mouse cells in an artificial cell, in an artificial membrane, and you had proteins from human cells. And they can easily be distinguished by antibodies. And they fused those two membranes together. And if the proteins remain static, then you should see half of the cell with all of the mouse proteins on one side and all of the human proteins on the other side. But after a minute, the proteins were evenly distributed throughout that hybrid cell, which allowed researchers to conclude that proteins are capable of freely diffusing through the membrane. There was another experiment where there were cells that were labeled with fluorescent antibodies. So these antibodies, these proteins, would fluoresce. And then at one point of the cell, they shined a laser, and that caused the fluorescent molecule to react, and so it would no longer fluoresce. So after a certain amount of time, if there was no diffusion of those proteins, you should have a spot with no antibodies. But after a short period of time, the antibodies were equally distributed throughout that cell. Now, this is what they did in artificial cells. So in artificial cells, you have complete freedom of movement. But when they looked at this in real cells, they noticed that some proteins can freely diffuse, and some proteins are completely static. And some proteins can only move in a straight line back and forth, and some proteins move only within a certain region of the cell. And so this allowed researchers to conclude that there are other factors at work with the proteins. And so in the absence of any restrictions, proteins are free to diffuse. But there are many ways that a cell can restrict the movement or direct the movement of proteins within the cell. And most of that is going to involve proteins and other molecules that are in the cell cortex. So a protein can be linked to the cell cortex, which will restrict its movement. A cell can set up what's known as fences. So it can create domains within the cell. And that can either be where you have these proteins that are immobile, and then you have a protein that is able to move within that region. Sometimes a protein will be linked to a cytoskeletal element, and it can move in a unidirectional fashion along that cytoskeletal element. In the case of tight junctions, these are tight junctions. We're going to look at this system again in just a little bit, so you should pay attention to this. These are intestinal cells. They line these are intestinal epithelial cells. And so here, this is the lumen of your intestine, where everything you eat and drink goes through. And then this is the other face of those cells, which is going to lead to the lymph and the blood. And so you don't want any of the fluid going through your intestine to be able to make it to your lymph without first being filtered through the epithelial cells. And so you have these tight junctions, which are large protein complexes that very tightly seal those intercellular junctions. So no fluid should be able to pass between those tight junctions. Okay? When you have these tight junctions, the proteins also can't diffuse past the tight junction. Okay? So those are effectively maintaining a domain here. So this face of the membrane can have a unique protein composition from this face of the cell. And we're going to talk about that when we talk about secondary active transport. All right, so I'm assuming you already know about osmosis and diffusion, right? Diffusion is simply the movement of molecules from a high concentration to a low concentration. It can be facilitated by a protein if those molecules cannot pass through the lipid bilayer. But if you're moving from a low concentration to a high concentration, that's active transport and requires some energy source. The diffusion of water is known as osmosis. Okay? I'm not going to talk about isotonic, hypotonic, and hypertonic, but you need to know that. So you need to go review that from an introductory biology book if you don't know isotonic, hypotonic, and hypertonic. Okay, so just a real quick 50-cent simple diffusion lecture. What's going to go through the plasma membrane without being facilitated by a protein? Things that are small and hydrophobic. Okay, the smaller and the more hydrophobic, the faster they'll diffuse. So things like oxygen, carbon dioxide, nitrogen gas, these will all simply diffuse through the plasma membrane relatively quickly. All right, then if you have a polar molecule but it's small enough, it'll probably diffuse, but it'll be slower than those hydrophobic molecules. So really, water will diffuse, but it's relatively slow compared to something like oxygen. And so if the cell needs to change the water concentration quickly, water will move through a protein. But water can certainly diffuse through the plasma membrane. Things that are larger and polar, like sugars, like glucose, or amino acids or nucleotides, these are polar molecules that are relatively large. They are not going to move through the membrane. They're going to need to go through a transport protein. And then anything with a charge, even protons that are teeny, 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 tiny, they have a charge, they will not go through the plasma membrane. Okay? They'll need a protein to facilitate their diffusion. All right, there's different kinds of proteins that are transport proteins. They're always going to be transmembrane proteins because they have to get from one side of the membrane to the other. There are channel proteins, which are basically just holes through the membrane. Okay, they have a very fast flow rate, but they're not very specific. Okay, they're pretty specific. You know, like a chloride ion won't go through a proton channel, but they can't, for instance, differentiate between L-glucose and D-glucose. Okay, whereas a carrier protein can. But a carrier protein is pretty slow. Okay, a carrier protein doesn't have a fast flow rate, but it's very specific, and it can also be used in secondary active transport. So a carrier protein is capable of harnessing the energy of diffusion in order to actively transport another molecule. Now, not all of them do, but carrier proteins can do that, whereas channel proteins can't. Channel proteins can open and close, but only transiently. Okay, so even if they don't have a stimulus that says open up, they may sometimes be open. All right. I'm not going to talk too much about ions and the electrochemical gradient because I'm assuming you already know about the electrochemical gradient, right? It's composed of two parts. The charge, whether negative charge is moving to a region that's negative or whether a negative charge is moving to a region that is more positive, right? So a negative charge will be attracted to positive regions and repelled by other negative regions. Okay, so this electrical component, in addition to the concentration gradient, which is simply the number of molecules on one side of the membrane from another. Okay, and these two forces work together and can produce, they can either work together so that the electric and the concentration are moving in the same direction, or they're moving in opposing directions. All right, so I'm assuming you already know about membrane potential and the electrochemical gradients. And so ion channels are going to capitalize on the electrochemical gradients, maybe for cell signaling or maybe for what do you call it? Energy transfer. So they're going to allow ions to move through quickly. And usually it's going to, well, I don't want to say usually. I mean, even potassium ions have ion channels. So it's only for diffusion, though. It's going to move with its electrochemical gradient. All right. Now, ion channels are able to open and close. So there's different types of gating. The gate is the region of the ion channel that will open or close. So the gates are classified based on the stimulus that opened them or closed them. But I want to stress that the gates are they're always some function of probability. Okay, so even in the absence of stimulus, there's going to be some small probability that they'll just open up. So maybe without stimulus, they'll be closed 90% of the time, but 10% of the time they'll still be open. And then if the stimulus comes, then 90% of the time they're going to be open, but still they're going to be closed 10% of the time. Okay, so it's just changing the probability that the gate will be open. So there's three different types of gating. A voltage-gated ion channel responds to membrane potential. So if the membrane potential of the membrane changes, then it will open up. A ligand-gated ion channel opens up in response to the binding of a molecule. Okay, so for instance, receptors. There'll be a receptor, it'll bind to a signaling molecule, and that'll cause the ion channel to open up. There's also mechanogated ion channels, and those are going to be things that respond to mechanical stimulus. So, for instance, the hairs in your ear, they respond to sound waves. The sound waves actually vibrate the hairs in your ears, which cause the ion channels to open up. Pretty cool, huh? This one, actually, a lot of people want to think that the Venus flytrap is mechanogated, but actually, this is a voltage-gated ion channel. So when the bug steps on it, it changes the membrane potential, and that causes the ion channels to open. All right, so these systems can very rapidly affect the signal from one side of the cell to the other, from one side of the membrane to another. And so, for instance, this is the system in your heart. So usually, or this also works for muscle cells, um, except not with sodium, with calcium. Usually this begins with a ligand-gated ion channel. So there's signaling molecules, and they bind to a ligand-gated ion channel, and that causes the channel to open, and the ions rush through. And once that initial cellular signal from one spot on the cell starts the membrane potential changing, the membrane potential change will open up voltage-gated ion channels that will then cause ions to rush through, and then that's going to change the membrane potential next to those gated ion channels, and now they'll open up, and now the ions will rush through, and now the membrane potential here is changing, so now these ion channels are going to open, and so you can see how that signal is going to be propagated from one side of the cell to the other. All right. So this is false because remember it's a function of probability. There'll be some probability that the ion channel will be open or closed. My baby's crying. I think somebody else will take care of her. We'll see. All right, so those are channel proteins. Remember, channel proteins have a fast flow rate, but they can't be harnessed. The energy of diffusion can't be harnessed, and they're not as selective as carrier proteins. Carrier proteins, they have a slower flow rate because they have to change their conformation, but they're very selective, and they can be coupled to other processes. Uh-oh. All right. So carrier proteins can be either active transport or facilitated diffusion. And so the glucose carrier is thought to randomly fluctuate between open to the inside of the cell or open to the outside of the cell. And so just like simple diffusion, it's a function of probability. If there's a high concentration of glucose outside of the cell, it's more likely that glucose will bind. If there's a low concentration of glucose on the inside of the cell, a glucose molecule might bind into the binding site, but it's not as likely. It's a function of probability. Okay, so diffusion tends to occur. Okay, whereas in active transport, there's an energy source, and that's going to allow the transport of molecules from a region of low concentration to a region of high concentration. Okay, there's two types of active transport. Primary active transport, the energy source is ATP, and in secondary active transport, the energy source is the diffusion of another molecule. So this is also known as coupled transport. Okay, in order for secondary active transport to occur, primary active transport must have occurred first in order to have built the concentration gradient in the first place. All right, so let's look at the sodium-potassium ATPase, or the sodium-potassium pump. This is one of the most well-studied pumps, and this is what animal cells spent almost 1/3 of their ATP on. It's very important maintaining the osmotic balance of the cell and also for using the sodium in secondary active transport, which we'll talk about soon. Okay, so this is a primary active transporter. It's transporting 3 sodium molecules outside of the cell, and it's transporting 2 potassium molecules inside of the cell. Now, both sodium and potassium are moving against their gradient. So while this is coupled transport, it's not secondary active transport because both sodium and potassium are moving against their gradient. And so in order for this to occur, ATP is required as an energy source. So, how come my GIF isn't going? Oh, there it goes. So if you look at this little animation down at the bottom, there's a very specific sequence of events that must occur. And the next step in this cycle won't occur until the previous step has occurred. And in this way, it ensures that the molecules are moving the way they are supposed to move. So let's look. The first thing that happens is sodium binds from the inside of the cell into the binding site. Then ATP is used to phosphorylate the pump, which causes a conformational change. So now the pump is open to the outside of the cell, and sodium leaves, potassium enters. Once potassium enters, the pump is dephosphorylated. And in its dephosphorylated state, it opens up to the inside of the cell once more. Okay, let's look at that again. I'm going to let it go through one cycle. Okay, it's going to open to the outside. Sodium leaves. That allows the potassium to enter their binding site. The potassium binding triggers the dephosphorylation of the pump. Now, the dephosphorylation of the pump is the conformation where it's open to the inside of the cell. Okay, then the potassium leaves. Now the sodium can bind. Sodium binding triggers ATP phosphorylation. Okay, so ATP phosphorylates the pump. The phosphorylated state of the pump is open to the outside. Now sodium can leave, potassium can enter, and potassium binding triggers the dephosphorylation of the pump, which triggers the conformation where it's open to the inside of the cell. Okay, so each step depends on the previous step. Sodium binding triggers ATP phosphorylation. The phosphorylated state of the pump is open to the outside. Sodium leaves. Potassium enters. Binding of potassium triggers the dephosphorylation. The conformation of the dephosphorylated pump is open to the inside. Potassium leaves. Notice the change of the binding site of potassium because potassium has the concentration gradient for potassium is minimal. There's very little difference between the inside and the outside of the cell for the electrochemical gradient of potassium. So potassium on the inside of the cell isn't capable of binding inside of the pump. Okay? All right. Another example of primary active transport is the calcium pump. And this is a really cool, they've done a really nice job characterizing this pump. This has a similar cycle to the sodium-potassium pump, but instead of the pump becoming phosphorylated, actually the entire nucleotide, the entire ATP molecule binds. So you have the calcium binding domain here. This is where calcium enters, and then when the pump changes conformation, it gets squirted out there. You have the ATP or the nucleotide binding domain. This is where ATP is bound, and then it is hydrolyzed into ADP and a phosphate, and then it's released. Okay, so I think I have a GIF of that as well. So here we have the nucleotide binding domain and the calcium binding domain. And you can see here the calcium enters, changes conformation, and it goes to the inside of the cell. Calcium enters, changes conformation, and goes to the inside of the cell. But I want you to notice there's actually 3 conformations here. Okay, calcium can enter. Calcium enters. Okay, then it changes conformation, and then it changes conformation again to squirt it out. Okay, that's because it's in one conformation. So calcium enters the binding site, and that triggers ATP to bind. Then ATP is phosphorylated, and then the calcium can be released, and then the nucleotide and the phosphate is released, and it is returned to its original conformation. Okay, so there's 3 conformations for this pump. The binding of ATP, after ATP has been hydrolyzed, and then when there is no nucleotide bound at all. All right, so we have these concentration gradients being built with primary active transport and the energy from ATP. Once you have a concentration gradient, you can use that concentration gradient to transport other molecules, such as glucose. So here we have the electrochemical gradient of sodium moving from the outside of the cell to the inside. Right? We've created a high concentration of sodium ions outside of the cell using the sodium-potassium pump. Okay? We are creating a sodium gradient. So the inside of the cell is more negative, the outside of the cell is more positive, so the electric component of the electrochemical gradient pushes sodium into the cell. And also, we are creating a high concentration of sodium outside of the cell using the sodium-potassium pump. Now, glucose, on the other hand, is at a higher concentration inside of the cell because we're importing glucose into the cell. But in the intestines, we want to remove glucose from the lumen of the intestine and bring it inside of the cell because we want to use the glucose for energy. And so we're going to use the energy of the diffusion of sodium down its electrochemical gradient in order to transport glucose into the cell against its electrochemical gradient. Now notice that we're talking about 2 things here. There's the physical direction of movement. The physical direction of movement is sodium moving from the outside to the inside and glucose moving from the outside to the inside. So this is a symport because both molecules are moving from the outside of the cell to the inside of the cell. Both molecules are moving in the same direction. The sodium is moving from the outside of the cell to the inside of the cell. The gradient of the electrochemical gradient is the opposite. Sodium is moving down its electrochemical gradient from a region of high concentration to a region of low concentration, while glucose is moving against or up its electrochemical gradient from a region, from a region of low concentration to a region of high concentration. So it's moving against its gradient. Sodium is being transported passively, whereas glucose is being transported actively. So glucose is undergoing secondary active transport. The energy for the transport of glucose comes from the diffusion of sodium. There is energy in diffusion. Energy, so you're changing potential energy into kinetic energy here. The potential energy of the electrochemical gradient of sodium is going to be harnessed. Okay? So this is what happens in animal cells. This also happens in plant cells, except instead of sodium, plants and bacteria pump out protons. Okay? So here we have primary active transport of protons using ATP. So the energy of ATP creates the proton gradient outside of the cell. Now, the plant is going to transport sucrose inside of the cell, for instance, into the phloem cell. And it's going to use the proton gradient in order to load sucrose into the phloem cell. So the energy of the diffusion of protons, we're building an electrochemical gradient. That's potential energy. Now we're going to use that potential energy to transport sucrose against its gradient. Both protons and sucrose are moving from the outside of the cell to the inside of the cell. So this is a symport. But the sucrose is moving against its gradient, and the protons are moving with or down their gradient. Okay? All right. Let's look at the transport of glucose from the intestinal lumen. Right? You just ate candy bar, so there's a high glucose concentration inside your intestine. And we want to get that in the bloodstream so we can use it to make ATP in all of our cells. Okay? So remember, the first thing that has to happen in this cell is we need to pump sodium out of the cell. So we're going to have a low concentration of sodium inside of the cell and a high concentration of sodium outside of the cell so that we can use that sodium gradient to transport glucose into the cell. Okay? Now remember, tight junctions can be used to set up membrane domains. Okay? So we're going to have tight junctions here and here so that we only have these glucose sodium symporters in this face of the cell. So we can transport glucose from the lumen of the intestine into the cell. Right? We don't want to transport glucose from your lymph into the cell because we're trying to get it into the lymph or into the blood. So we'll only have the glucose sodium symporters in the face of the cell that is in contact with the lumen of the intestines. So now we're creating a high glucose concentration inside of the cell because we're actively transporting glucose into the cell using secondary active transport because we already made our sodium electrochemical gradient with the sodium-potassium pump. Okay? Now, the sodium-potassium pumps won't be restricted to any one face. They'll be all over the cell. But on the face of the cell that is in contact, what they call the basal domain, it's actually probably not directly in contact with the lymph, but eventually it gets there after several layers of cells. We're going to move the glucose from a region of high concentration inside the cell to a region of low concentration outside of the cell, right? Because the glucose is being taken away by the lymph or by the blood. So there's low concentration outside of the cell, a high concentration inside of the cell because we're actively transporting it into the cell with the sodium glucose symport. So is this going to be active transport or passive transport? This is going to be passive transport because we're going from a region of high concentration to a region of low concentration. All right, so this is going to be that glucose facilitated transporter. All right, so this is very elegant. It takes into account all the things we talked about before. The sodium-potassium pump first creates the sodium gradient by primary active transport. Then we create a high glucose gradient inside of the cell using secondary active transport with the sodium glucose symport. And now that we have that high concentration inside of the cell, we can just let the glucose diffuse out of the cell by facilitated diffusion. Isn't that beautiful? All right, so what are the names of these proteins again? Which one maintains the sodium gradient? The sodium potassium ATPase, or the sodium potassium pump. Which protein makes the high glucose concentration? That's the sodium glucose symport, or the sodium glucose cotransporter. It's not an ATPase, no ATP necessary. All right, so that's it for membranes. I will try to have the aerobic respiration lecture up. Ooh, I want to say soon. Soon, I promise. All right? Bye, guys. Have a happy new year.